The next speaker is trying to bring epithelium and fibroblast together, Kevin Kim from the University of Michigan, who's going to talk about epithelial-fibroblast interactions in lung fibrosis. Kevin.
All right, thanks. I want to thank the organizers for giving me a chance to really present this sort of classic topic, unlike some of the other topics that we've had at this meeting, like telomeres and stem cells that are newer and perhaps more exciting. This is one that's, depending on how you look at it, sort of an older topic or one whose importance has withstood the test of time. And I want to do something a little bit different, which is talk a little bit about my own research, but there have been several prominent papers recently that challenge a lot of the dogma around epithelial-mesenchymal interactions during fibrosis. So I wanted to highlight some of that work as well. And I do want to stick to a sort of tried and true outline that many of the reviews on epithelial-mesenchymal interactions during fibrosis follow, which is to spend a little bit of time talking about epithelial-mesenchymal interactions during development, spend most of my time talking about different pathways involved in fibrosis, and then end with a little bit on epithelial-mesenchymal transition. So I think all of you know that pulmonary fibrosis is a multicellular process. There's injury and damage and activation to the alveolar epithelial cells. There's destruction of the alveolar basement membrane. There's accumulation of activated fibroblasts and deposition of a fibrotic, collagen-rich scar. And all of these processes don't happen sort of independent of each other, but are coordinated through cell-cell interactions, with one cell producing a cytokine or a growth factor or matrix protein that then activates a receptor on its target cell, leading to a pro-fibrotic phenotype. And so epithelial-mesenchymal interactions are prominent in this interaction during fibrosis. And a lot of what we know about epithelial-mesenchymal interactions in the lung are derived from studies of lung development. So you have these classic lung development studies where you can culture embryonic epithelial cells alone, or co-culture them with mesenchyme, and the mesenchyme secretes factors which allow the epithelial cells to proliferate and undergo rudimentary branching and differentiation. Some of the factors that are secreted by the mesenchyme include FGF10, because if instead you use a bead soaked in FGF10, it again allows the epithelial cells to proliferate, undergo branching that is reminiscent of branching morphogenesis during development. And a number of prominent pathways have been worked out, and these are some of them here. And the communication goes both ways, both from epithelium to mesenchymal and mesenchymal back to epithelium. And some of the prominent pathways that have been worked out include the fibroblast growth factor family, Wnt beta-catenin, hedgehog, the TGF-beta superfamily, which includes BMP and activins, and a extracellular matrix integrin pathway are all prominent. And so one pathway that's sort of, say, a clean pathway in epithelial-mesenchymal interaction is the hedgehog pathway, because hedgehog in the lung is expressed exclusively on the epithelium, and its receptor is expressed exclusively on the mesenchyme. And this, in general, is thought to lead to mesenchymal cell proliferation through a pathway that involves smoothend and glee. And this feeds back to FGF10 that signals back to the epithelium. And as you would expect, if you delete sonic hedgehog, you get an underdeveloped lung. But really demonstrating the complexity and the need for regulation of these pathways, if instead you overexpress hedgehog, you also get an underdeveloped hypoplastic lung. So fibrosis is not development, but in IPF you have these areas of fibroblast foci These are areas of active matrix deposition with accumulation of activated fibroblasts and myofibroblasts, and there is this overlying layer of atypical cuboidal hyperplastic epithelial cells shown in these two figures here. So maybe this is an important site of epithelial mesenchymal transition, important for fibrosis. A lot of studies of pulmonary fibrosis really focus on fibroblasts, but we know from familial causes of fibrosis that Lisa will talk about later that epithelial cell dysfunction can cause pulmonary fibrosis. So, for example, mutations in epithelial-specific genes such as surfactant protein C and surfactant protein A2 are sufficient to cause pulmonary fibrosis. And even in non-familial pulmonary fibrosis, as I mentioned, there's clear evidence of epithelial cell apoptosis, epithelial cell injury and dysfunction, and overall activation. And I think the idea is that with injury, be it from oxidative stress or DNA damage or other factors, that diverse injuries to the epithelial cells or the epithelial cell response to diverse injury can converge on this secretory phenotype, and we've heard in the morning session about a senescence-associated secretory phenotype. So diverse injuries can converge on epithelial cell secretion of pro-fibrotic factors, with the idea being that some injury leads to epithelial cell apoptosis, but also activation of epithelial cells to secrete a number of pro-fibrotic factors, which then can signal in autocrine and paracrine fashion, leading to mesenchymal cell activation and proliferation. Many of these factors, importantly, unlike hedgehog, are not epithelial cell specific, but perhaps the epithelium is an important or necessary source of some of these pro-fibrotic factors. We showed a number of years ago, for example, that if you delete CTGF, and we've seen a number of potential therapies for CTGF discussed at this meeting, that if you delete CTGF specifically within epithelial cells, you can block bleomycin-induced fibrosis, suggesting that the epithelial cells are an important and necessary source of CTGF. So perhaps in addition to blocking the activity of CTGF, epithelial cells might be an interesting target for blocking production of CTGF. So this is a listing here of a number of pro-fibrotic factors that epithelial cells can produce. And again, they're not the only source of many of these factors, but they're an important or necessary source for the outcome of the fibrosis. So this is where I am in the talk. I wanted to highlight three separate pathways that in a lot of ways challenge the old dogma with regards to epithelial mesenchymal interactions in fibrosis, including TGF-beta, hedgehog, and growth factor receptor tyrosine kinase signaling. So the first is TGF-beta, which is known to induce many pro-fibrotic phenotypes and is the most well-studied inducer of lung fibrosis. This is just a sketch of the TGF-beta pathway. Importantly, it begins with a secretion of TGF-beta in a latent or inactive form by epithelial cells, but also by many other cell types. This binds to the extracellular matrix through this latent TGF-beta binding protein and is activated by a number of integrins binding to an RGD motif. This allows it to bind to its receptor and lead to a SMAD-dependent and independent activation of its target cells, leading to the phenotypes described in the last slide. So initially, this seemed like it was a pretty straightforward epithelial to mesenchymal story with the main integrin that was initially described, alpha V beta 6, which in the lung is only expressed on the epithelial cells. And again, this binds to an RGD motif on the latent TGF-beta, allowing it to bind to its target cell, which was mainly thought to be fibroblasts, leading to fibroblast activation and fibrosis. Beta-6 integrin knockout mice are protected from fibrosis, and there's upregulation of beta-6 integrin in IPF. So it seemed like a very clean epithelial cells as the site of TGF-beta activation, and fibroblasts as the main source in terms of response to TGF-beta. Several integrins can bind to an RGD motif. This is a diagram just summarizing all of the integrin dimers. All these ones here in the blue triangle are RGD binding integrins, and importantly, most, if not all, of these integrins can lead to TGF-beta activation. And many of these, unlike alpha V beta-6, many of these other integrins are expressed on multiple cell types, including fibroblasts and myofibroblasts. Just dovetailing on what Dan Shepperland was talking about, there's a lot of interest in matrix rigidity in terms of not just the response to TGF-beta, but in TGF-beta activation. So the idea is that TGF-beta binding to a stiff matrix on one side and contractile force on the other side leads to greater TGF-beta activation. So perhaps if you have a contractile cell like a myofibroblast, you could see greater TGF-beta activation. And work from Dean Shepard has confirmed this, showing that if you delete the alpha V subunit in just in myofibroblasts, and again, this knocks out a number of these RGD binding integrins, you see protection from fibrosis shown here and shown histologically here. So both epithelial cells and mesenchymal cells are required for TGF-beta activation during fibrosis. And the same is true for the response to TGF-beta. So this is two separate papers where they deleted the TGF-beta receptor in fibroblasts in this case, and epithelial cells in this case. And in both cases, what they saw is protection from fibrosis, shown histologically here and quantified here. So really the data support the notion that with regard to TGF-beta signaling, both epithelial cells and fibroblasts are required for both the activation and response to TGF-beta, consistent with this notion that there is a complex epithelial mesenchymal interactions requiring both cell types. So TGF-beta and fibrosis could be a topic on its own, but I wanted to move on to hedgehog signaling since it's so prominent in epithelial mesenchymal interactions. It hasn't been studied as much in the context of fibrosis, and as I alluded to, it's mainly been studied in the context of lung development. And again, hedgehog produced by epithelial cells leads to mesenchymal proliferation through the smoothened Gli signaling pathway during lung development. So perhaps there's a reemergence of this during fibrosis. And you heard earlier today that there's a drug targeting the hedgehog pathway. And I think something like this is a paper that came out a few years ago, is what a lot of us believed, was that after injury, this is a Gli reporter mouse as an indicator of hedgehog signaling. After injury, you have upregulation of Gli indicating an upregulation of hedgehog. Here, if you block sonic hedgehog, you don't attenuate bleomycin-induced fibrosis, but if you add in extra hedgehog, you see more bleomycin-induced fibrosis, suggesting that at least in part, hedgehog has a role in leading to mesenchymal cell proliferation and accumulation during fibrosis. Really, in a surprise report from Ed Morrissey's group, they found almost the exact opposite in a couple of ways. First, they found really high levels of sonic hedgehog, even at baseline in the adult lung, uninjured state. This is the green GFP here, high levels of hedgehog both in airway epithelial cells and in alveolar epithelial cells. They found high levels of Gli, again indicating activation of the hedgehog pathway in mesenchymal cells under uninjured conditions. The other way it differed was that here, instead of hedgehog inducing proliferation, it's actually suppressing proliferation. So this high level of hedgehog signaling is suppressing proliferation. When they delete hedgehog, again uninjured, it actually releases this quiescent state and allows for mesenchymal cell proliferation. These are all these red cells in Ki-67 staining. And what they really argue, again, this is the opposite of what a lot of us thought, is that there's high levels of hedgehog signaling. With injury, including with bleomycin, there's a dramatic downregulation of hedgehog signaling, and this downregulation of hedgehog signaling releases the mesenchymal cell quiescence and secondarily releases an epithelial cell quiescence. So inhibition of hedgehog leads to fibroblast proliferation and secondarily epithelial cell proliferation, leaving you with either fibrosis or resolution, depending on which wins out. So really a lot of interest in this classic epithelial mesenchymal pathway, and I think a lot of sort of work that needs to be sorted out. And then last, I wanted to move on to a growth factor receptor tyrosine kinase pathway focusing on FGF. And this is because, as you all know, we have two therapies which can slow the progression of IPF. One of them is Nintedanib, which is a nonspecific tyrosine kinase inhibitor. There has indeed been a lot of interest in tyrosine kinase inhibitors as potential therapy for IPF. Nintedanib blocks the fibroblast growth factor receptor. It blocks platelet-derived growth factor receptor, which are both receptor tyrosine kinases, which are clearly involved in fibrosis. The FGF-FGR pathway clearly has a number of pro-fibrotic activities, including, as its name suggests, fibroblast growth, but also fibroblast activation. But the FGF family is large and diverse. There's some 20 different ligands and 4 different receptors that all signal in different ways. And there are a number of reports, including this one from a year ago, suggesting that some of these FGF pathways can be anti-fibrotic. So this is a report from Martin Kolb's lab showing that FGF1 FGF1, for example, can attenuate TGF-beta-induced pulmonary fibrosis. This is shown histologically here. This is control lung, TGF-beta-treated mice with increased fibrosis, and TGF-beta-treated mice co-stimulated with FGF with inhibition of fibrosis. And this was through inhibition of fibroblast activation. This is fibroblasts expressing smooth muscle actin after TGF-beta and blocked by co-expression of FGF, and by inhibition of epithelial mesenchymal transition with epithelial cells, upregulation again of smooth muscle actin, and blocked with FGF. And indeed, in this review, a number of FGFs have been shown to be pro-fibrotic, but a number have also been shown to inhibit fibrosis. And there was a poster here about FGF2 inhibiting pulmonary fibrosis as well that didn't make it on the slide. And so a number of these FGFs can block fibrosis through inhibition of EMT or fibroblast activation, or stimulating epithelial cell proliferation. FGF7, also known as KGF, has been studied for years as a potential therapy for acute lung injury and perhaps fibrosis in the setting of lung injury. So it's a really diverse family, and clearly part of the way Nintedanib works is through inhibition of the FGF-FGR pathway, but further clarification and dissecting this pathway I think is really needed to understand the epithelial mesenchymal interactions of FGF during fibrosis. So that's a good segue into epithelial mesenchymal transition. This is the ability potentially of epithelial cells to become mesenchymal-like with inhibition of some of their epithelial features and acquisition of some of their mesenchymal features such as migration, invasion, and production of fibrotic matrix. There was interest for a while that perhaps fibroblasts are not the only source of activated myofibroblasts, but there may be other cell types, including fibrocytes and epithelial cells, and this is important because epithelial cells are so prominent in the uninjured lung. If some of them transition into an activated cell type, they might account for a decent portion of the activated cells. And it's also important because there could be perhaps different signaling pathways leading to activation of these different cell types. And so the two questions that we had is, does EMT occur during fibrosis, and is it important for the fibrotic process? And first you have to ask, does it occur during fibrosis, because it can't be important if it doesn't at least occur. So the first question was, does EMT occur? And it had been proposed that the gold standard method for demonstrating that EMT occurs is to use a fate mapping strategy. This is using an epithelial-specific Cree crossed with a lock-stop-locks reporter mouse, and then see if any of these permanently labeled epithelial cells express markers of activated fibroblasts during injury. The problem is that multiple groups use this gold standard technique with very different results. So for example, in the context of lung fibrosis, 4 separate groups have shown, using this fate mapping strategy, that epithelial-derived cells can express markers of activated fibroblasts, but at least one prominent group showed no evidence of EMT. And this is true for kidney fibrosis and liver fibrosis as well. So there is definitive evidence that epithelial to mesenchymal transition occurs, and there is definitive evidence that epithelial to mesenchymal transition does not occur. And this isn't just true for EMT. So this is true for pericytes as well. There's a notion that pericytes are a progenitor for activated myofibroblasts. And here, I think the difference is the most stark. There is clear definitive evidence that pericytes are not a source of activated myofibroblasts, and yet there is clear evidence that pericytes account for up to 70% of the myofibroblasts that develop in fibrosis. And the same is true with fate mapping strategies for fibrocytes as well. Clear evidence both for and against. I think at least part of the problem is in how we define EMT. So you can have a very rigid definition, which is to say that epithelial cells must lose all epithelial traits and gain expression of all mesenchymal traits. They must become indistinguishable and homogeneous. And one author describes it as a true stable conversion of one differentiated lineage into another. And I would argue, almost from a semantic basis, that makes EMT during fibrosis impossible. You can have a loose definition, which is maybe too loose, which is to say that epithelial cells are dynamic. They have more than one physiologic program, and after injury, they can downregulate some of their homeostatic functions and proteins, which are no longer useful, and upregulate a repair process that overlaps in some ways with mesenchymal cells. And so I think the really important question is, do epithelial-derived cells acquire functions that we have traditionally attributed to activated myofibroblasts, rather than saying, does EMT occur or not? And for me, the sort of the hallmark of an activated myofibroblast, it's not as simple as this, is can they express and produce collagen in a meaningful way during the fibrotic process? So this is old data showing that epithelial cells can express pro-collagen. You can do this in a fancier way. We did this in collaboration with Paul Walters, who I think has left. But you can laser capture these atypical epithelial cells that overlie the fibroblast foci and do transcriptional profiling of them, and you see marked upregulation of type 1 collagen. And then more recently, this is from Jeff Witzett's group, you can do single-cell RNA-seq. So you can, at a single-cell level, get the RNA transcriptional profile by RNA-seq of alveolar epithelial cells from idiopathic pulmonary fibrosis. they found several different categories of epithelial cells, but at least one category had marked upregulation of type 1 collagen by this RNA-seq method. And then we took a different approach using transgenic mouse model to say, what if we specifically delete the gene for type 1 collagen? And we found marked reduction of bleomycin-induced pulmonary fibrosis. In these mice, really demonstrating an important role for epithelial cell-derived type 1 collagen in this model. And our question now is whether the type 1 collagen itself can act as a signaling molecule important for epithelial-mesenchymal interaction. So this is my last slide. I see Z is prompting me to finish. Pulmonary fibrosis really involves a number of complex epithelial-mesenchymal interactions. I think we've uncovered many of the prominent pathways, but there is still much to sort out with the complexity of these interactions. Thanks.